to
Recording in progress.
that they can use to accelerate that they've used to accelerate repurposing strategies for their patient communities. Before we get started for today, I just want to go over a couple of housekeeping items. First, presenters will be sharing presentations for this, and we will be saving time at the end for audience Q&A, but we encourage you to enter your questions into the chat feature, and we will go to the questions at the end of the webinar, and we'll go through as many as we can, time permitting. Secondly, we will be recording this webinar and posting the recording on the website following its completion. And thirdly, we will ask that you please mute yourself through the duration of the webinar. We are doing this webinar in Zoom meeting format to provide a more interactive opportunity for participants. Next slide, please. Oh, actually, go back. Could you go back to Train? So Train, for those of you, well, most of you are part of Train, but if you're not, you're probably part of Rare's One. So as part of Train, we have more than 160 foundations that are engaged, and we do opportunities throughout the year to help support patient organizations' capacity to fund and advance biomedical research, connect with each other, and leverage their collective voices for biomedical research. Next slide. We have, for those of you that are not part of this, we have rolled out Train Communities of Practice last year, and they are accepting members on an ongoing basis. And Train Communities of Practice are a peer-to-peer sharing opportunity for members that participate in our capacity building program. So other patient organization leaders sharing promising practices, strategies, and ideas that address topics that are of greatest interest to them. We have 4 communities of practice organized around the domain areas found in our research partnership maturity model, which include expertise, which might focus on things related to board management, internal staff, patients, which can focus on topics engaging patients in research, collecting patient data for research purposes, as well as developing your research strategy. A one on money, so talking about topics related to fundraising and approaches for funding research, and finally relationships, which can talk about all the different partnerships you might need in industry, academia, and other partners to help advance your research. Meetings take place quarterly, and we encourage membership is on a rolling basis, and we'll share the chat. We'll share in the chat the link to join up in that community of practice if you haven't yet engaged. Next slide. So coming up for next year, we just want to share some updates. We have some exciting opportunities that will be rolling out in 2025 to help with more opportunities to help patient organizations think about some emerging areas for patient engagement that they might consider, including how to engage with CMS in the access determination space, as well as how maybe some resources and opportunities to engage in the medtech space, which includes devices, diagnostics, and digital health products. So stay tuned for more information on upcoming programming for those opportunities, as well as other topics. In addition, Train turns 20 years old next year. So as part of that, to celebrate, we are going to be rolling out the Train Impact Series, which will be highlighting members of the Train and other capacity building programs to share their stories about the impact and role that patient organizations and patients can play in research, as well as to talk about how participation in some of our programs has impacted them. So we're very excited to roll this out, and you'll be seeing more from us on our social media channels and in our newsletters about Train's 20th year. Next slide, please. Finally, I just wanted to mention that Faster Cures will be participating in the World Orphan Drug Congress to share a miniature interactive session around our research partnership maturity model, which is a framework that organizations can use to think about how they can grow as research partners. So we'll be doing a brief session to have organizations apply the model to think about where they'd like to grow strategically as an organization for research purposes. We've done a couple of longer format workshops over the past few years, and it's a great opportunity that we'll be doing at that workshop. So if you haven't yet had a chance to attend the larger workshop and you're going to be there, we'll love to see you participate in that. Next slide, please. So without further ado, I'm going to turn it over to Kristen, who will be moderating our conversation and introducing our speakers.
Wonderful. Thanks so much, Ray. Good to see everybody. Thanks so much again for joining us here at the end of the year for an evergreen topic, repurposing, repositioning, reprofiling products. David introduced us to a new term just now, which is resurrecting shelved assets. So all of these terms have been used to describe taking a therapeutic asset that has been researched and developed for one condition and applying it to another. As Ray alluded to, it can offer a less expensive and more rapid pathway to approval, and is as a result of great interest to a lot of players in this biomedical innovation ecosystem, including patient communities. It does, however, have a lot of challenges, including financial, liability, intellectual property, data access issues, you name it. So its deployment as a strategy has been a little challenging, shall we say, over the years. But we really felt like this was a great time to take up this topic again, because there's some new news, there's new assets, new platforms and AI technologies that are enabling faster and better matching. There are new partners for development. There's new approaches to getting patients access to these repurposed or resurrected products. And we have great examples now, like we're going to hear from today, of patient organizations that have effectively partnered to help navigate these challenges. So without further ado, we're going to get started with our presentations. First, we're going to hear from David Fajenbaum, who is currently the co-founder, well, among other things, he wears a lot of hats, is co-founder and president of EveryCure, which is one of those platforms that I alluded to. Many of you know him also as the founder of the Castleman Disease Collaborative Network, where he kind of piloted this this strategy on himself. And we'll hear a little bit about his personal story, but really excited to hear about the development of EveryCure and how the groups on this call might be able to benefit from and partner with it. Turning it over to you, David.
Well, Kristen, thank you so much for that intro, and just such an honor to be a part of this amazing panel and to have the opportunity to share about the work that we're doing. I also see that many of the participants in today's webinar are dear friends and colleagues in this repurposing and rare disease world, so it's really great to be with everyone. So just to sort of frame things, I think it's important to talk about where we are in biomedical research. And of course, each of you that's part of a rare disease group or a more common disease group, you understand that there's work to be done for your condition. But I think it's also helpful for us to take a step back and to say, how are we doing generally at drug development? I think we as a society should be really proud that there are over 4,000 FDA-approved drugs that are approved for about 4,000 human diseases, which is really just a medical marvel that we've done that. However, there's still over 14,000 diseases that don't have a single approved therapy. And in our current system, we spend a lot of effort and resources to develop new drugs for those 14,000 diseases without any approved therapies. But the reality is that in many cases, there are existing medicines that could be repurposed, and not in all cases, but in many cases. This is a very personal topic for me. I was a third-year medical student at Penn when I became critically ill with a horrible disease called Castleman's. I spent months in the ICU. I had my last rites read to me, and I received a bunch of chemotherapy that kept saving my life, but I kept relapsing. And going through this journey made me realize that those chemotherapies weren't made for my Castleman's, but they kept saving my life. So maybe there was some other drug out there that maybe also wasn't made for Castleman's that could maybe save my life. And so that set me off on a really simple journey, which was figure out what's going wrong in me and see if there's something out there that could maybe fix me. And I did that work both in the laboratory at the University of Pennsylvania and through starting a foundation called the Castleman Disease Collaborative Network that's now over 12 years old and doing really important work. And between the lab work and the CDCN, we uncovered that an old drug called sirolimus could potentially be effective. And that's because we discovered that a particular communication line in the immune system called mTOR was turned into overdrive. And I'm just showing a couple pictures here of a typical lymph node. Brown is positive. It means that communication line is turned on. Blue is negative. It means it's turned off. And the first image is a typical lymph node. The second one's my lymph node. And so with all the brown staining, all the mTOR staining, that made us think that an inhibitor of that communication line called sirolimus could maybe treat me. And so we began testing this mTOR inhibitor on me, which is approved for another rare disease called LAM. I see my friend Vera's on the line from the LAM Foundation, could maybe be useful. And that ended up working. And I've been in remission for over 10 years. And if I'm fortunate enough to make it to January 5th of next year, that'll be 11 years of remission. So during this 11-year remission, I joined the faculty at Penn and started this Center for Cytokine Storm Treatment and Laboratory. And we've gone on to uncover many more repurposed drugs. So now we're a total of 14 drugs for diseases they were not intended for, helping thousands of patients all over the world. These are some of the many that are alive thanks to repurposed drugs. And I just want to share a couple examples. So this one's of Michael. Michael was diagnosed with metastatic angiosarcoma in 2016 and was failed to respond to the 2 chemotherapies that are used for it. We did something very simple. We looked in the literature and we found that a paper published 3 years earlier suggested that a PD-1 inhibitor, or that PD-L1 expression was increased in angiosarcoma. So we thought maybe a PD-1 inhibitor could be useful for Michael, though it had never been used before for his form of cancer. We confirmed increased PD-L1 expression in his tumor in 2016 and started him on therapy as the first patient that we're aware of with angiosarcoma to receive a PD-1 inhibitor. And he's doing great over 8 years. April would mark 9 years of remission. And because he's done so well, this drug has been given to many other angiosarcoma patients all over the world, and now it's recommended first-line therapy. And I love this example because it required no brilliance at all. It required doing a simple PubMed search, finding work that had been done 3 years earlier by a great lab, but no one had actually taken that work in the lab and translated into patients. And that's something that obviously is so critical. And just a couple months ago, Michael walked his son down the aisle at his wedding, an experience that he never thought that he would be able to have over 8 years ago. Thought I'd share about Allie's experience. She has a rare condition called DATA2. Her father's Chip Chambers, who many of you all will know in the rare disease community. DATA2 is a horrible condition where kids die from having hundreds of strokes before they reach their teenage years. And a researcher about 20 years ago made a connection that researcher thought a TNF inhibitor could be a useful treatment approach for DATA2. But about 15 years went by where TNF inhibitors were being used by some, but not others. And frankly, lots of children were dying from DATA2, even though the world knew a TNF inhibitor could be useful. There was clinical data supporting it, but the treatment guidelines weren't out. The utilization of this TNF inhibitor for DATA2 wasn't widespread until Chip did the incredible work to establish these treatment guidelines. So this is another great example where even a step beyond the previous example where the lab data was there, but the clinical data was not. That's angiosarcoma. In this case, the lab and the clinical data was there for DATA2, but the utilization was not there of this drug. So every time I walk past a pharmacy, all I think about is how many drugs are there that could be helpful for other patients, for other diseases that we don't even know about right now. My cure was sitting on the shelf for years while I was in and out of the ICU, and we've now been able to do this a number of additional times. So I launched a nonprofit called EveryCure along with Grant Mitchell and Tracy Sikora almost 3 years ago that's on the mission to unlock the full potential of each and every drug to treat each and every disease that drug possibly can. That doesn't mean that there's a cure for every disease, but it does mean that every drug out there that could be useful for another disease, we believe should be used for that additional disease. And we do this by utilizing biomedical knowledge graphs, where we apply a handful of different artificial intelligence algorithms, basically on the world's knowledge, to quantify the likelihood of every FDA-approved drug, all 4,000 of them, to treat every human disease, all 18,500 of them. So it's a total of about 75 million calculations, which, of course, until recently would never have been possible. But thanks to the incredible work done by the translator program at NCATS to build the fundamental ingredients, which are biomedical knowledge graphs, fundamental algorithms, we are actually able to do this. And so again, just a huge shout out to the incredible work from Translator to build these fundamental building blocks, which our group at EveryCure is utilizing to then optimize these algorithms and really create a fit for purpose biomedical knowledge graph. We're going to be sharing all these scores in a little bit over a year for the world to look at every drug versus every disease. But we internally at EveryCure, we use these scores to then determine what are the things at the top, what are the drug disease matches that have the highest scores. And then we look deeply. We have a medical team that goes deeply through each one of those opportunities that says, okay, this is a great opportunity, but we need more lab data, or this is a great opportunity, we need to do a clinical trial, or this is a great opportunity, we just need to get the word out to more people. So we evaluate the things at the top, we pick the greatest opportunities, we do the work that's necessary, and our goal is always to ensure that the drug is fully utilized for every patient that can benefit, it's covered by insurers, and in some cases, we work with the FDA to actually get a label change, but that's not in all cases, because in fact, today, 30% of all prescriptions in the US are off label. So lots of off label prescribing happens. For many of these old, cheap generic drugs, there's actually no path to getting a label change. And so we do whatever we can to make sure drugs are utilized. And so I want to share quickly some archetypes that we put repurposed drugs into. So we call frontier explorers, or what we call repurposing opportunities, where there's a glimmer that this drug might work, like there was with sirolimus for Castleman's. We've got some lab data, but there's no clinical evidence. There's no clinical adoption. These are ones where we like to do more laboratory work to really better understand, why would sirolimus work for Castleman's, for example. Another one is what we call clinical gems. So in this case, the mechanism is pretty strong, and we're ready to get into clinical use, but there's more clinical work that's needed. So pembrolizumab for angiosarcoma, that was an example after Michael received it, where there's some clinical data, but we need to gather the clinical data together. We need to maybe launch a clinical trial or build a registry. So more clinical work is needed for clinical gems. And our last category we call unsung heroes, or like a TNF inhibitor for DATA2, where the mechanism's strong, the clinical data is strong, but patients just aren't using it. So there's more work that needs to be done to get the drug to more people. And of course, you don't need to do that if the drug's on patent, because there's sales forces that do the work. But once drugs go off patent, there's no one to push forward the use of a drug like this. We ran the first version of our algorithm almost 2 years ago, and we're really excited by the results that we got back at EveryCure. Of course, you can imagine that we looked across the whole list of everything at the top, and interestingly found propranolol for ovarian cancer was right up at the very top, but also looked within specific diseases like Castleman disease, and we're really interested to see the number one and the number three top predicted drugs for Castleman's were TNF inhibitors, which had never been used before for Castleman's. But it was very interesting because right around that time, a doctor had been taking care of a patient and considering putting that patient onto hospice care. This is the letter that Luke wrote, indicating that his patient would need to go onto hospice care. And right around that time was when we got this really high prediction for a TNF inhibitor, and recommended that this patient, Al, actually receive a TNF inhibitor for the first time that we're aware of for Castleman's. It saved Al's life. This Christmas will mark, or I guess in the new year, will mark about 2 years that he's been in remission. And we have a paper that's now been accepted in the New England Journal of Medicine that describes this work. More recently, earlier this year, I had this patient, Joseph, who has POEM syndrome. He was also at the end of his road. Nothing seemed to be working, and we ended up recommending 3 drugs that scored well across 3 different algorithms for Joseph, and he responded really, really well to therapy, and of course left the hospital and was back with his family. So what I described just now is what I like to call, looking for the lowest hanging fruit on a given disease. So we're focused on POEM syndrome or Castleman's. What's the best treatment for that disease? And our algorithm is good at that, and that finds the lowest hanging fruit for Castleman's or POEMs. But what is really transformative about what we're doing with EveryCure is not looking for the lowest hanging fruit in a single tree or a single disease, but actually looking for the lowest hanging fruit across the entire forest. And so when you generate a normalized score for all drugs versus all diseases, you can say, what are the greatest opportunities to help people? Not necessarily people with one disease or another disease that you focus on up front, but what are the greatest opportunities to help people? And that helps you to uncover things like leucovorin for a rare subtype of autism, where kids have autoantibodies against the folate receptor, or maybe arginine for sickle cell disease, or bosutinib for ALS, or lidocaine for breast cancer, or metformin for long COVID. And so it's by looking across the entire forest and using an algorithm that can generate a normalized score for every drug versus every disease that allows you to find the greatest opportunities to help people, full stop. And so what comes next after we generate the scores? Well, it's working with patient organizations, disease organizations, insurance companies, regulators, governments, to make sure that these drugs are advanced as far as they possibly can to reach as many patients as possible. So for those of you that are part of a patient group, We are going through this prioritization process on a monthly basis, and if there's a drug disease match that scores highly and it's involves your disease of interest, we will reach out to you and we'll want to know from you, what do you think about this drug? What do you think about the evidence? What do you know about this drug for your disease? The other thing you can do is you can actually proactively reach out to EveryCure at everycure.org/insights, and you can tell us if you know about a repurposed drug that you think our medical team should evaluate. So that's right now how we're working with patient groups. We're not currently taking on requests to work on a specific disease or another disease out of the 18,500. We're basically letting the data tell us what drug disease matches to pursue, but we are not currently able to say we're going to focus on Castleman's or you name the specific disease. We look for matches. We've got some amazing partners listed on the screen who've been supporting us from the very beginning, but we really do need your help. I shared about this one opportunity for sharing insights, and hope you will we'll reach out if you have ideas for repurposing opportunities, or if you have any thoughts on ways that we can help more patients more quickly. Thank you guys so much.
Fantastic. Thank you so much, David. Incredible presentation, and a lot of content to unpack there. And hopefully, I will encourage folks to put questions in the chat as you're thinking about them. You don't have to save them for the end. But I'll just say one thing, which I didn't say at the beginning of David's presentation, which is one of the things that really kind of caught our attention when he first started going down this path was not just the technology. It's clearly a great technology platform, and maybe a better AI algorithm than anybody else has got to do this kind of matching. And thank you also for the shout out to NCATS, who we'll hear from in just a bit, that helped form the basis for some of that. But it's just this recognition that maybe the biggest part of the challenge isn't the technology part, isn't the matching part. That's a huge challenge, but it's the what happens next part, and the driving it through creation of guidelines, or there's just like a number of different pathways that you could go down to do this, and I don't think anybody has really been exploring those in as robust a way as you all are doing. So really exciting work. We're going to turn next to hearing from Marianne Clancy, who's Executive Director and Senior Director of Strategic Partnerships at Cure HHT. And Marianne's going to tell us a story that gives me anxiety sometimes when I hear about it, because it's like one relatively small group just taking on an enormous risky task, and with really quite remarkable success thus far on the repurposing, or I'm not sure which term you would use, repositioning, reprofiling, resurrecting front. But I'm going to let Marianne tell us the story about what her group has been doing recently, and I think a lot of you will be able to see yourselves, hopefully, in the work that they're doing. Take it away, Marianne.
Thank you so much, and thank you to Kristen and Faster Cures, and also sharing just this panel with both David and Christine, who are so esteemed and I have respected for such a long time. And it's a real privilege of mine to take you through our journey at Cure HHT, a small advocacy organization that truly has gone through the repurposed and repositioning of therapies for our patient population. Next slide, please. So what is HHT? I'm just going to take you through very quickly a couple of intro slides. It's a genetic autosomal dominant disease that impacts the development of blood vessels, primarily capillaries, that form direct connections between arteries and veins. And in these direct connections create in small blood vessels, such as the nose and the GI tract, telangiectasia. Think of them as tangled vessels that can rupture and are prone to bleeding. When these vessels are in the larger organs, such as the brain, the lung, the liver, these malformations are quite large and can certainly lead to catastrophic hemorrhage of the brain, the lung, high output heart failure due to liver, and development of pulmonary hypertension. And it's due to cell mutations in cell signaling pathways that are responsible for blood vessel growth. And primarily 3 genes have been known since the mid-90s. 3 genes are involved. 2 genes, endoglin and ACVRL1, are primarily responsible for about 90% of HHT. HHT affects about 1 in 3,500 people, nearly 2 million people worldwide. And then a third gene, SMAD4, which is HHT in juvenile polyposis, that creates colon cancer in young children and adults, as well as HHT. So structurally, these vessels are unstable and they're prone to bleeding. It's not a problem with the blood or the ability to clot. We oftentimes say it's the wine bottle and not the wine. So each family has their own mutation in one of these genes, and it's autosomal dominant, meaning that every child that is born to a parent has a 50-50 chance of getting this gene. This disease was first described in 1896, and to date, there still is not one FDA-approved therapy, like so many of us. Next slide, please. So here's a normal capillary bed, and as you see, arteries carry oxygenated blood away from the heart to the organs, to the tissues of the body. And you see a nice capillary, and then you see veins, and these are vessels that carry deoxygenated blood from the organs to the heart, where they pick up oxygen and then are carried to the body. Next, could you just, this is a build. This is what an arteriovenous malformation in a telangiectasia looks like. So you really create a hypoxic situation. Next slide. So most blood vessels in HHT have a normal structure, but a small number of them are abnormal. And the signs and symptoms really depend on the number and the location of where these vessels are. And we are still learning why some of these vessels become abnormal. Next slide, please. So HHT is diagnosed according to what's called the Curacao criteria, primarily because there was a research meeting done on the island of Curacao. And so if 3 of these are present, 3 or more, you have a definite HHT, possible HHT if 2 are present, and unlikely if less than 2. Primarily, it's characterized by spontaneous nosebleeds that are recurrent, and that occurs in families. Telangiectasias are these tiny red spots that can appear on the skin, the mouth, the face, the hands, as well as certainly internally. And then internal lesions, these AVMs that occur in the lung, the brain, the liver, GI, and spinal cord, and certainly with the family history of first-degree relative with HHT. Next slide, please. So at Cure HHT, we were formed in 1991, like all small nonprofits start with a group of patients and an interested physician. And our goal here is advocating for patients, advocating for care and therapeutics, and transforming lives. At the same time, we estimate that only 3 of 10 patients are diagnosed with HHT. So our goal is educating patients, families, referring them for care, opening centers of excellence, which we have done. We invest in research, fund young researchers, partner with biotech. And really our goal here is to accelerate some breakthroughs and collaborate with our research network. Next slide, please. So we started in 1991 with 1 center of excellence. This is a very complex disease with several organs involved. So you need a multidiscipline team that can really help to diagnose, certainly screen and treat patients, because if you're screened and treated, you can live a long, healthy life. If you're not, you can be prone to catastrophic events. So we have 32 centers of excellence in North America and another 20 globally, 20 plus, all throughout Europe, Asia, a few in South America. We have a lot more work to do, though. Next slide. So HHT is first diagnosed, believe it or not, between the ages of 27 and 45 years old. And that's because nosebleeds are fairly common. But if they are in your family and you're having several a day, several a week, that is abnormal. Although most people with HHT think that's normal for their family. 28% are diagnosed after the age of 46. Believe it or not, we had an 85-year-old that was diagnosed. Oftentimes what will happen is there's a family history. Sometimes 20% present with stroke, and no one's putting together some of these events such as stroke or sudden death with chronic bleeding and anemia. Next slide, please. So at Cure HHT, we created this research network in looking at patient priorities. Where are our gaps and what are our recommendations? Next slide. So I'm going to talk to you a little bit about a journey that we went through 15 years ago. We had a family actually from Philadelphia. This gentleman was in his 60s. He was a dentist. And he was incredibly anemic. People with HHT walk around with hemoglobins of 7 and 8. And he was bleeding chronically from debilitating nosebleeds, very, very weak. And there was some, his son was a physician, pediatric ophthalmologist, and thought about, gee, there might be this drug Avastin that's really helped with macular degeneration. It's an anti-VEGF drug, which is what happens with HHT. We have too much VEGF. Let's see if this might be helpful. Found a physician in Southern California who would consider giving this drug off-label to the dad. So off they went to San Diego, and there was a first case report of using Avastin to treat HHT, and lo and behold, the vessels got smaller, the bleeding was reduced, and patient got a lot better, had a normal hemoglobin and elimination of anemia. Since then, there were several other papers. Then more physicians started to use this drug off-label, and they started publishing the data. Some were used for high output heart failure. Again, more publications of using it for chronic bleeding and anemia. And then in 2019 at our scientific meeting, since we have all these centers of excellence, you know, we had physicians who were using it off-label, Avastin, and they were using maybe 20 patients were on the drug and getting results, in another center maybe 10, in another center 30, until we said, you know, at Cure HHT, we really need to get your whole team together, these teams together, and do a multi-center publication of what we're seeing in terms of results. And that was the INHIBIT trial. That was nearly 200 patients, and it was published in 2021. And that is something that at Cure HHT, we encourage them to do, because we've been hearing from patients, certainly on social media, and those patients that we talk to that would contact our office, that this treatment off-label was life-saving. I needed no more blood transfusions. I can live a normal life. It reversed my anemia for the very first time in my life. So what we did is we provided educational webinars and news to our patient community. We collaborated with our centers, certainly to gather their experience. And we funded 3 very small pilot trials that were positive. And that all resulted, lo and behold, in 2000, I believe it was 18, we obtained CMS approval for using Avastin, bevacizumab, for HHT treatment. So the company wasn't interested in funding any trials for HHT, despite all this evidence, but it's continued to be used today, and it's saving a lot of people's lives. Next slide. So that led to Cure HHT advocating for additional treatment, looking at different additional types of drugs, because Avastin and bevacizumab is useful for many patients, but not everyone could tolerate it. Some had side effects, and some it wasn't working. So in 2018, we were able to get a U54 grant, and which led to a multi-site clinical trial with the PI at Cleveland Clinic. And Cure HHT is an advocacy organization, was very involved very early. We collaborated with the PI in trial design, site selection, developing the patient-reported outcomes, validated quality of life instruments. We prepared written letters and emails promoting the trial, evaluation of lists, referring patients to the trial, to the centers that were participating in the trial, involved in the monthly conference calls with all of the sites and preparing quarterly progress reports. Next slide, please. So this was the overview of the pomalidomide trial, known as the PATH trial. We had 11 active sites. We had 177 patients who were consented. And I will tell you, patients really were so desperate for treatment. They've been bleeding to death, multi-generations of people for years and years and years. We will do a webinar and we'll gather 500 people at a webinar that is discussing a clinical trial for HHT, because there had been nothing up until most recently. So we had 144, this was as of November in 2022, and 70 people had completed. Next slide, please. Okay, so I'm just quickly going to go through. We looked at reduction of endpoint. We looked at reduction of nosebleeds, reduction of iron infusions, reduction of blood vessel, red cell transfusions. Next slide. So basically, it was 4 milligrams of pomalidomide over a period of 6 weeks. This trial was closed early after 144 patients went, primarily because there was a statistically significant improvement in hemoglobin, reduced nosebleeds, reduced transfusion and iron infusions. I'm happy to say that it was published in the New England Journal of Medicine in 2024. We were really thrilled with that. And I'm going to end with the last few slides here because I'm almost at time. Next slide, please. The pazopanib story was such that, and I'll go through this very quickly. In 2015, GlaxoSmithKline funded a multi-center, we thought, proof of concept trial. It was for a drug that was approved for kidney cancer, pazopanib, known as Votrient, at 800 milligrams. It was repositioned at 50 milligrams for HHT. 7 patients completed the trial. They showed improvement across hemoglobin, nosebleeds, and GI bleeding, and it was halted because the whole portfolio was sold to another company. Next slide. Long story short, what this shows you is that we decided to take it forward ourselves. We obtained access to the generic compound. We invested $100,000 over 6 years, wrote an IND, got an FDA breakthrough designation. We also wrote grants at the same time and received $6.2 million for clinical trial. We outsourced everything from a CRO to device companies to statisticians. And we anticipate completing enrollment for 80 people by the end of next month. And if all is well, we anticipate that this drug at 75 milligrams for treatment in HHT, we hope to take it to FDA for its first approved treatment. Next slide. We can skip that. Next slide. So I just want to end with this. In 2015, we just had bevacizumab, a few drugs, and then from 2018 to 22, we had 3 drugs, all repurposed. Next slide. This is where we are now going forward, 24 to 28, 2028, all types of different therapeutics, sirolimus, tacrolimus, and several others to be repurposed for HHT. Next slide. Lessons learned: be relentless. Taking a drug to market is not for the faint of heart. Know your value and that of your community, because your family's life depends on it, and those in your community. Collaborate and be sure to have a seat at the table. The patient voice is very powerful, and be sure you're heard. Thank you so much.
Incredible. Thank you, Marianne. You really did get to the heart of the matter there at the end, and the source of all my anxiety is just that incredible story with pazopanib. So hopefully people will have questions. Please put them in the chat. And if Marianne can answer them in the chat, I'm sure she'll be happy to. But I did want to make sure we get to Christine Koldis, who's Director of the Office of Drug Development Partnership Programs at NCATS, the National Center for Advancing Translational Sciences, which, don't tell anybody, is FasterCure's favorite center at NIH. And I'm sure you'll hear a little bit about why that's true. They have built incredible resources for this whole ecosystem across a variety of fronts for many years, but many which David's already kind of cited as foundational for this repurposing work, and they support the efforts of a lot of rare disease organizations and research as well. So I'm going to turn it over to Christine for her presentation about what's available at NCATS these days that's of interest on this topic.
Right, thank you so much. Yes, thank you for the invitation and for the very kind introduction to NCATS. What I want to do today is actually just give you a taste of some of the types of things that we do to help with drug repurposing, along with other things. I think a lot of people here probably know that NCATS is a disease agnostic organization, and what we mostly focus on is developing technologies and methods and processes that are going to make the process of translating from preclinical or non-clinical space into the clinic and hopefully ultimately to marketed treatments. So let's go to the first slide here. Today I'm just going to cover a couple of things in these 4 different categories. One is how we could use clinical observation for drug repurposing. The next is computationally assisted identification. Thirdly, sort of the more traditional high throughput screening type approach that we have here at NCATS. And then I'll let you know about preclinical development, just sort of, more generally. So on the next slide, you'll be seeing these various different things. These are the little icons that I'm using for each of these. You may want to have your phones ready. I have peppered my presentation with QR codes where QR codes will actually work. But next slide, we will start with talking about Cure ID. So many of you probably know Heather Stone at the FDA. Cure ID was really her brainchild. She had been in the infectious disease space where she was seeing a lot of cases of infectious diseases that were essentially rare diseases where there wasn't an approved medicine, particularly if we go to less fortunate parts of the world. And so what doctors would resort to, the same as a rare disease doctor would do, is to just prescribe things off label, try what you can based on whatever it is that you know. I think Heather felt really frustrated at the same time that seeing all of this happen and that information, there wasn't really a way for people to share their experiences with these drugs, whether it's the physician, whether it's the patient. There was no way to sort of capture that information and disseminate it. So she had approached NCATS, our intramural program, about 10 years ago to help her to develop a tool, an app that would actually allow people to share this kind of information. And on the next slide, you'll see that in recent weeks, months, you go to the next slide, or maybe there's just a delay. This is really starting to take off now. Public launch happened just before COVID, rather fortuitously, I suppose. And so it hasn't been, in the public domain for that long, but now we're really starting to see the uptake. And Heather's recognizing that, this obviously has potential well past infectious diseases. She'd really like to get to vulnerable populations, such as rare diseases, pediatric populations, pregnant women. These are populations where this could be potentially really, really useful to be able to share the information without necessarily having to go through the work that it would take to actually put out a traditional publication. So on the next slide, I actually have the QR code here. If you want to get into Cure ID, learn more about it, start to play with it, just scan that QR code. So on the next slide, we'll talk about translator, which already came up today. David gave us a really nice shout out. And we have what we are doing in the translator program is, I think of it as a knowledge aggregator. So we are trying to pull together about 200 different sources of knowledge, meaning, primarily databases. We're still pretty heavily reliant on literature, information that's in literature. But what we want to do is be able to help researchers. It doesn't have to be for repurposing specifically, but to help researchers find connections across data. And we sort of started with connections to drugs. And so, one of the searches, if we go to the next slide, you'll see that this little drop down that you sort of see in the middle there just shows we have just 5 different types of queries you can do. One is what drugs could treat a condition. And then the other 2 relate to chemicals that can regulate a gene up or down, or genes that are affected by a particular chemical. And again, this is intended really to help researchers, but we also felt that what drugs may treat a condition would be really helpful for people who are in the rare disease space. So now I'll give you a quick example of how this has been useful in real life. On the next slide, the Hugh Call Precision Medicines Institute at the University of Alabama, which is led by Matt Might, they have been using Translator for a while now, much like David, trying to identify potential treatments for refractory cases or for rare or ultra rare conditions where there is no treatment available. And I think it was a couple of years ago, maybe just 1 year ago, they had a family come in where their young daughter had a condition called Shine syndrome. This is very, very rare. It's actually so rare that we don't have the disease Shine syndrome modeled in Translator. We didn't have the ability to search for what drugs might treat Shine syndrome. But because it's a rare disease, as you all know, that means that we know the gene that is actually mutated. This happens to be a haploinsufficiency mutation, meaning that this little girl has one good copy and one bad copy. And so what we did was we searched Translator for, or they, I should say, searched Translator for what drugs could upregulate this gene in the hopes that that good copy, if they could make more copies produce more of the good copy of the gene, that they might be able to help her with these symptoms. And so guanfacine came up, which is really interesting because it is a hypertension treatment that has been around since the early 80s. It had more recently was approved in an extended release formulation to be administered to kids for ADHD. So her clinical team, looking at this and realizing it's been around for a long time, been in a lot of people, pretty good safety profile, has actually been in children for ADHD, led them to the conclusion that this would be a relatively low risk thing, and it would be worth trying considering how devastating these symptoms are for her. So on the next slide, you'll see that after just 5 months of being on guanfacine, you see in this picture on the right-hand side, the left side of that picture is what her coloring looked like before she was taking guanfacine. And just 5 months after being on guanfacine, she really was able to improve her motor skills considerably. And her mom said that social behavioral skills had also improved as well. And so this was just off-label prescribing of the drug for a single individual. So next, we will shift to the NCATS pharmaceutical collection. This is an NCATS intramural program. If we go to the next slide, you can pick off the QR code there so you can learn more about this. So what NCATS Intramural Group did is they've actually amassed 3,000 of the approved drugs as well as experimental drugs, so that they're in one library for high throughput screening. And so this is obviously a very popular library for screening. It's really the lowest hanging fruit. These are things that have already been in the clinic. If you were to identify a potential new use for it, this would be the best way to sort of fast track to going into the clinic for a clinical trial, or potentially even for, an N of one type of situation like I just described for Shine syndrome. So on the next slide, I wanted to show you, going along with that, is that in our intramural program, we're very different than any other intramural group at the NIH. Most intramural labs here are very much like academic labs. PIs have their own research interests and they pursue those. At NCATS, what we do is we help support other people's research, and the NCATS pharmaceutical collection is an example of that. So in order to get access to it, you would submit a proposal saying, here's the condition, here's what we have, here's what we need. And that goes through a review process. And generally what we're looking to is the collaborators usually coming to us with a target or a probe, some sort of early stage compound, or they have subject matter expertise and specialty resources, such as an assay or animal models or other type of model system. And it's really the collaborator who's going to have the therapeutic hypothesis. NCATS really has the know-how and a lot of specialty technologies for actually helping people to develop their drugs. So we will revisit this model a little bit later in the presentation. So on the next slide, now I'm going to switch gears to the preclinical development opportunity that we have right now. Next slide. This one. This QR code you probably really want. This is the only one so far, or the only one really in my presentation where there's actually money available, right? The other ones are truly resources, potential for collaboration or what have you. But this one is actually a funding opportunity. And this is really for somebody who already has a lead compound, but they're not quite ready to go into the IND enabling studies because they need to still demonstrate proof of concept in some kind of an animal model or some kind of a model system. So this is specifically to sort of help bridge that gap. The next receipt date is going to be this coming June. And this will support efficacy testing. It could be to repurpose something. It could be for a novel agent. It doesn't have to be for drug repurposing. You do need to have a compound in hand, and you need to have an established rare disease model for this. We unfortunately do not support rare cancers. It's purely sort of a resource thing for us that NCI has a lot more money, and we really want to be able to spread the wealth as much as we possibly can. And then if awarded, you would be required to establish a steering committee or an oversight committee to just make sure that we keep the patients in the loop and are really addressing things according to what their needs would actually be. And along with this, I wanted to go to the next slide to tell you about the AstraZeneca Open Innovation Program. So let's say you have your model, but you don't have a compound yet. Take a look at their Open Innovation site. They have compounds both that are clinical stage compounds as well as preclinical stage compounds that are, well-developed things that they do make available. There's still a process, again, because it does cost the money to make these available. There's a proposal process, but I think it's a pretty low bar for you to submit to request access to these compounds to begin to explore them. So that could go really well. We're really well hand in hand with this preclinical proof of concept idea or funding opportunity. And then on the next slide, this is where I want to switch back now. So just told you about a funding opportunity, switch back to our collaboration model on the next slide. Should you make it through that and you have your proof of concept is already done, and for a rare disease, our Therapeutics for Rare and Neglected Diseases program, the TREND program, with QR code here, has a bunch of different capabilities and know-how. This is this, I think, is really the type of thing that's particularly difficult to find in an academic setting. It's the kind of thing that you might go to a CRO for or something like that. This, again, is done through our collaboration model. So you wouldn't get money, but you don't have to pay money either. It's truly done as a collaboration. And they are very, very good at what they do, and very, very successful. Great group to work with. So I definitely encourage you to look into that. And then on the last slide, if you missed all those other QR codes, this one will take you to a two-page sort of overview sheet of drug repurposing at NCATS. And a lot of the stuff that I talked about today has links to it from this QR code. So that is it. Thank you very much.
Oh, fantastic. Thank you, Christine. That was a great way to kind of wind up this series of presentations. So many great resources and opportunities there, and incredible ways that NCATS supports this whole topic of repurposing and this community of patient organizations. I know we've only got a few minutes left, and folks can continue to pop questions in the chat, and we'll try to follow up with them afterwards. We will be sharing the slides with the permission of the presenters, and links. We'll make a list of the links that folks alluded to or provided. But I did want to ask one closing question, if I could. It's a big question, but it kind of follows on Katrine's question in the chat about intellectual property. I mean, one of the things that has just interested us about these, some of the approaches that you all have been describing, is that it's a little bit of an end run around the traditional system of drug development and marketing, right? And intellectual property plays a huge role in that. And kind of going, directly to guidelines and off-label prescribing and payment, CMS, trying to get CMS to pay for things off-label. But I'll just ask kind of the open question, maybe starting with you, David, kind of what you're thinking about kind of the role of intellectual property here, owning it or controlling it. How important is it? How much should we be indexed on that at this point?
It's a great question. I think in some cases you do need it, but in the vast majority you don't, especially if we're talking about generic drugs. I mean, if you're not changing the dose or the formula and the drug is inexpensive, insurance companies tend to cover inexpensive drugs. They don't like to cover expensive drugs, but they tend to cover inexpensive drugs. And so if you generate good clinical evidence, like I said during the talk, 30% of prescriptions are off-label. So doctors are prescribing things off-label all the time. Insurance companies are paying for things all the time, but we have to generate the evidence, and it doesn't. And so in that case, I don't think you really need IP if it's generic. If you need to change the dose or the formula, you need the IP. But I think that if it's on the market, I think that it's just about generating the evidence.
Mm-hmm. Great. Marianne, Christine, do either of you want to comment on the intellectual property conundrum?
I tend to agree with David. I think that, we've been really successful, and again, we're taking pazopanib forward just because it's different dosage, and for our community we had no approvals whatsoever, so that's why we're taking it forward. But I think for the most part, it would not be necessary. And people are using off-label now all the time on propanolol. There's another drug that they're using that I didn't mention, but certainly a number of them.
Yeah, I would say unless you needed a new formulation. For example, it's only a tablet, and your patient population wouldn't do well with having to swallow a tablet. It wouldn't be worth making the investment to get a use patent, for example, for something that's already out on the market and that's available as a generic.
Yeah, great. All right, great closing thoughts. Lots of hope for moving these approaches and these ideas forward, and lots of great partners to do it with. So thank you, all three of you, for the great presentations, and to all of you for participating again here at the end of the year. Happy New Year to everyone. We look forward to more great discussions in 2025 with all of you, and hope you have a great holiday season.
Thanks, Chris.
Thank you.
Thanks,
Thank
everyone.
you.
Bye.
Thank you.
Bye-bye.
Thank you.